Okay, so welcome to this next video in which we are discussing the effects of sympathetic stimulation on the heart. Okay, so we've seen one of these complexes now by which we can activate the L-type voltage-gated calcium channel and increase the amount of calcium it's going to let into the cell during a calcium sparklet via the activation of the beta-1 receptor via noradrenaline. So we now want to see another example of a complex like this, which is going to allow type 2 protein kinase A to phosphorylate the type 2 ryanodine receptor. Okay, so I don't think I'll try and squash it in there, so we'll get another piece of paper. Okay, right. So, this next complex then. So again, what you have is your beta-1 adrenergic receptor. So here's the plasma membrane of our cardiomyocyte. Here's the beta-1 adrenergic receptor with its 7 membrane-spanning domains. Okay. Here's the heterotrimeric G protein, which is a GS heterotrimeric G protein. Okay. And this time, specifically in this complex, you have only adenylyl cyclase of the 5th type, so adenylyl cyclase 5. So here is our adenylyl cyclase 5 here. Okay, so this is adenylyl cyclase 5. Okay, and let me just add a bit of colour on, so we'll alter the colours around a little bit this time. So our beta-1 adrenergic receptor is in vivid purple here. Okay, and noradrenaline or norepinephrine, so we'll switch around the name of this time. NE, norepinephrine, is going to come and bind to our beta-1 adrenergic receptor. You'll often hear people write beta-1 AR for beta-1 adrenergic receptor. Okay, we'll have our alpha S subunit of this GS heterotrimeric G protein in green here. And then the adenylyl cyclase 5 we'll have in red here. Okay, with its 2 transmembrane regions. Here's TM2, with the 6 membrane-spanning alpha helices. And now, we need the membrane of the sarcoplasmic reticulum because I told you that this setup is aimed at phosphorylating the type 2 ryanodine receptor. Okay, so here is our type 2 ryanodine receptor, this massive great protein sitting in the membrane of the sarcoplasmic reticulum here. Okay, and we want to try and phosphorylate this. So, what we have basically is a protein, another A cap. Okay, so here's this other A cap here, which is bound to the adenylyl cyclase 5, and it's got to specifically be adenylyl cyclase 5 that it binds to here. Okay, and oops, what am I drawing here? Right, so let's have it going around like that somewhere. Okay, so it's specifically binding to adenylyl cyclase 5, and it's known as, it's got 2 names. It's known as M-A cap beta, okay, and that stands for the muscle-specific A kinase anchor protein beta. Okay, so I'll write that down somewhere. So this is the muscle-specific A kinase anchoring protein, A kinase, so remember A kinase is just another name for protein kinase A kinase anchoring protein, or ACAP. Right, okay, and it's the beta form of that, so muscle-specific A kinase anchoring protein beta. Now, it's got another name. Its other name is to call this ACAP6. Okay, so it's the 6th A cap, but often you'll hear it referred to as the MA cap beta more often. It's more often called that than it is referred to as the A cap 6. Okay, right, now this binds to the type 2 ryanodine receptor here, so this is our type 2 ryanodine receptor, RYR2. Okay, and I'll just make sure that it's obvious that it's a tetramer. Okay, and it also binds to another protein that's in the membrane of the endoplasmic reticulum. So we'll draw this protein here. And this other protein is a protein known as nesprin. Okay, so it binds to nesprin which is a protein in the membrane of the sarcoplasmic reticulum. And this just helps the muscle-specific A kinase anchoring protein to join the two membranes, basically. Okay, so let's colour this one in green. So here's the ACAP beta, the MACAP beta in green here. And it's bound to the type 2 ryanodine receptor, which we'll have in orange, I think. And then we finally need to show the protein kinase A of the second type bound to this, this ACAP protein, this MACAP beta. Okay, so we'll draw the protein kinase A bound here. Okay, so again, it's one of these type 2 protein kinase A's here. So it's got the second regulatory domain that allows it to bind to this ACAP here. If it was the type 1 and it had the type 1 regulatory domain, then it wouldn't be able to bind to the ACAP. It would instead therefore be soluble. Okay, so here are these two regulatory domains of this second type, so the R2 subunits. Sorry, I keep saying domains, I mean subunits. And here are these two catalytic subunits here, which are bound with the regulatory subunits in this R2-C2 complex. Okay, right, so let's colour these in. So these catalytic subunits here we'll have in yellow. Okay, if that's visible. And we'll have the R2 subunits now in pink here. Okay, there's one, and here's the second one. Now, what's going to happen here then? When you stimulate the beta-1 adrenergic receptor with noradrenaline, what's going to happen is that beta-1 adrenergic receptor is going to stimulate this GS, heterotrimeric G protein, which is then going to stimulate the adenylyl cyclase 5 enzyme, which will then produce cyclic AMP, which it will throw onto this type 2 protein kinase A enzyme down here. The cyclic AMP will bind in each one of the four binding sites for cyclic AMP on the regulatory subunits. The regulatory subunits will change their conformation, releasing the catalytic subunits from their grasp, and the catalytic subunits will then become active, and they will go and phosphorylate the type 2 ryanodine receptors. And when they phosphorylate the type 2 ryanodine receptors, they will increase the conductance of the type 2 ryanodine receptors. So they'll increase the amount of calcium that is released from these type 2 ryanodine receptors. They'll make them more sensitive, and they'll increase the amount of calcium that they release. So you'll get larger releases of calcium from the ER, which will mean that you get larger calcium sparks, which will mean that the calcium spike, if we now look at our calcium concentration versus time graph, okay, so this is calcium concentration versus time, then let's say initially what was happening in our cardiomyocyte was calcium was going up when the action potential arrived, so the L-type voltage gated calcium channel was opening, letting calcium in, calcium was going through and causing calcium-induced calcium release from the type 2 ryanodine receptors. That then leads to this rise in calcium. Then the action potential goes, so the L-type voltage gated calcium channels stop releasing calcium onto the type 2 ryanodine receptors. The type 2 ryanodine receptors stop working. Okay, so they close as well, and then what happens is the Circa pump is going to return the calcium back into the ER. Okay, then you're going to get an interval, and then you'll get an action potential again, and it will happen all over again. Right, now what we're going to do is we've phosphorylated the L-type voltage gated calcium channel, and we've also phosphorylated the type 2 ryanodine receptor. So what will happen this time is the amount of calcium that comes in through the L-type voltage gated calcium channel will be bigger, and in addition, the amount of calcium you release from the type 2 ryanodine receptor will have gone up, so the amount of calcium you're going to release will have got bigger. Okay, now the question is, how are we going to activate the removal of calcium as well? Because we said that we don't just need higher spike, we don't just want more calcium to be released, we also need it to be removed again. We need to increase the rate at which we're removing calcium so that we can make up for this, because we don't want calcium to be left in the cytoplasm at the end of the action potential. We don't want it to still be there. We want to relax completely back down. So we need to remove all the calcium out of the cytoplasm again. Okay, so we're going to have to activate the mechanisms for removing the calcium out of the cytoplasm and returning it into the sarcoplasmic reticulum. Okay, we haven't seen that yet. We're coming on to that. Okay, at the moment we've increased the size of our calcium spike. Okay, and I just want to add another thing. So I've told you that when the action potential ceases, then the L-type voltage-gated calcium channels will close. Okay, that's true. And that because of the lack of calcium from those L-type voltage-gated calcium channels, that will serve to help inactivate the type 2 ranitidine receptor. That's also true, but there's another thing that I need to add to that, which is that the other reason that calcium stops being released from the type 2 ranitidine receptor is that the type 2 ranitidine receptor has complicated kinetics, basically. It's both activated and inactivated by calcium. So if I have a graph here of the activation of the type 2 ranitidine receptor, so this is activation of type 2 ranitidine receptor, so RYR2. And on the x-axis we plot calcium concentration. Okay? Then at lower concentrations of calcium, you get a massive great activation of the type 2 ranitidine receptor. But then as you go to a higher concentration, it goes down again, like in this sort of bell-shaped curve, normal distribution. Okay, so at these really low levels of calcium here, where you're not getting much activation, that's simply because there's too little calcium to actually cause activation. Then as you increase the calcium, it activates the type 2 ranitidine receptor. So these are the sort of levels that the L-type voltage-gated calcium channel will be spitting onto the type 2 ranitidine receptor. But if calcium concentration goes too high, it inhibits the type 2 ranitidine receptors. So they have these dual kinetics as far as calcium is concerned. It can both activate at low concentrations and inactivate at high concentrations. So basically, when the L-type voltage-gated calcium channel opens and spits calcium onto the type 2 ranitidine receptor, that will, and I don't know why I was pointing at that, that's not the L-type voltage-gated calcium channel, but when it does, calcium will activate the type 2 ranitidine receptor to open and release calcium from the SR. But when the calcium concentration goes up and up, as it will because the type 2 ranitidine receptor is releasing this calcium, that too high concentration of calcium now in the vicinity of the type 2 ranitidine receptor, that will serve to negatively feedback on the type 2 ranitidine receptor to inhibit it. So that's also a really important reason for why you stop releasing calcium from the SR, basically. It's also because when the calcium concentration gets to a certain height, it will start inactivating the type 2 ranitidine receptor, and that's why you will stop releasing that calcium. So phosphorylating the type 2 ranitidine receptor with the catalytic subunits of the protein kinase A is going to increase the conductance of calcium through that type 2 ranitidine receptor. It's also going to potentially shift this curve a little so that it doesn't get as inactivated as soon, basically. So I've just told you that the calcium will negatively feed back and inhibit the type 2 ranitidine receptor. Phosphorylation by protein kinase A will reduce the sensitivity of the ranitidine receptor to that, so that it will inactivate at higher calcium concentrations than before. Okay, so both of these things are how the protein kinase A is going to activate the type 2 ranitidine receptor, so that overall we get larger calcium spikes in the cytoplasm of the cardiomyocyte. Okay, and we'll continue our discussion in the next video, where we'll talk now about how we're going to activate the Circa pump so that we can remove this calcium more quickly as well, so that we can still go down to a very low level of calcium in the interval.